Hello, let's talk about metabolic liver disease. I think the most important, most central concept you ought to know regarding this SDL is that alcoholic and non-alcoholic chronic liver disease appear the exact same way. They follow the same progression from fatty change into inflammation, into scarring, then potentially hepatocellular carcinoma. So they've got the same root pathology. Oxidation of fat generates an inflammatory response that you can see manifested as the presence of immune cells. And that inflammation engenders fibrosis, which is particularly mediated by stellate cells, remember, which can transform into myofibroblasts. And then there is a cancer risk once you hit cirrhosis. So let's begin by looking at those three particular stages of change. We've got steatosis here. That's fatty change. And it can be macro or microvesicular, referring to the size of those fat droplets. If you see a bunch of large fat bubbles, roughly many of them the same size as hepatocytes, that's a macrovesicular distribution. But if you see some smaller fat bubbles sitting inside hepatocytes, that's what we would call a microvesicular distribution. So if you just see fatty change, that's steatosis. When you see immune cells on top of fatty change, that's what we call steatohepatitis. And the itis part of that word refers to the inflammatory response. And inflammation is mediated acutely by neutrophils and then chronically by macrophages and lymphocytes. So when you see those cells in a liver biopsy, you know you're at least looking at an itis, whether it be acute or chronic. And you will start to see some fibrous change in response to all of that inflammation. And in this particular slide, it's this blue collagen that you're seeing. It's a trichrome stain, and collagen and other fibers pick up blue. So repeated attacks of steatohepatitis can lead to cirrhosis, the hallmark of which is nodularity. So islands of hepatocytes, nodules like globes, like grapes almost, surrounded entirely by blankets of fibrosis, which is still blue in this picture, and I've given it a dotted yellow outline. And you've got to have both of those for a tissue to be cirrhotic. Now, there's a whole other SDL on cirrhosis. You should be very familiar with it. In reference to this SDL, and anytime you see a picture of the liver, you need to start by picking out what stage of damage it's in. Because again, this is a pretty consistent progression. You're asking, is it fatty change? Is it inflammation? Or is it cirrhosis? So where I start, I look at a picture of the liver. I say, is this cirrhotic yet? And that's kind of obvious because there's so much collagen in cirrhosis that you'll right off the bat be able to say, yes, it's cirrhosis, or no, I don't think that's cirrhosis. I don't see a lot of scarring. So I start with cirrhosis and then I work my way backwards. I say, maybe it's not cirrhotic, but do I see immune cells? Do I see a lot of dense, small, dark nuclei, lymphocytes, macrophages, and neutrophils? Because if you do, that's a steatohepatitis, because the immune cells represent active inflammation and they're participating to damaging the liver. They're the source of a lot of liver injury, your own immune system is. And so if you don't see a lot of immune cells, you're continuing to work backwards and you're going to say, okay, there's just a lot of fatty change. This is the steatosis stage, which is commonly benign and is in fact reversible in many cases. So that histological progression, again, really important, is the same across alcoholic and non-alcoholic chronic liver disease. And you might take a biopsy of the liver and see a little bit of simple steatosis over here and some really bad cirrhosis over here. It's not exactly mutually exclusive. Now, the fatty change will begin around zone 3, around the central vein. I think that's important. Anytime you see zone 1 or zone 3, put it in your memory bank because potential test question right there. The fatty change is going to begin in zone 3, and I believe that's due to 2 primary reasons. Number 1, that's the lowest oxygen content of any zone of the hepatic lobule. And then number 2, the zone 1 hepatocytes, the periportal hepatocytes, have all your glycolytic enzymes. And so they don't really have their pro-fat enzymes upregulated transcriptionally to be constantly able to make or metabolize fat, whereas your zone 3 hepatocytes are not as glycolytically active. They're more reliant on aerobic metabolism. And so not only are they going to have more radicals due to that lower oxygen perfusion, they're also going to have more fat to start with because their enzymes are already up and running. Hope that makes sense. So simple steatosis, as we just looked at, pure fatty change with no immune infiltrate. As we get into steatohepatitis, we start to see an inflammatory response. And in alcoholic steatohepatitis, there are some characteristic lesions. Well, I wouldn't even say alcoholic because these same lesions are going to show up in non-alcoholic steatohepatitis. So if you see any of these following findings, even with a lack of active immune cells there, active inflammation, you're still going to say there has been some inflammation there. If you see things such as hepatocyte ballooning and Mallory bodies and fibrosis, bridging fibrosis, if you start to see those things, you're going to say itis, inflammation has already happened here. And then if you see the inflammatory reaction, like at the bottom of page 3 is describing here, neutrophils and T cells, then of course that's a hepatitis, not a steatosis. So what do some of these findings look like microscopically? Here's a picture of steatohepatitis as a result of alcoholic liver disease. And you see many immune cells, macrophages, lymphocytes, small dark nuclei that I've outlined in yellow. You also see some hepatocyte ballooning outlined in blue with some Mallory bodies starting to form within the cytoplasm of those ballooning hepatocytes. Here's another look at a steatohepatitis. We take a look at it. Do we see any fibrosis? Not really. So it's not cirrhosis, and it's pretty zoomed in too. Next step, do we see any inflammation? Yes, there is absolutely a big column of inflammatory cell infiltrate right there in the middle of that liver. So we see some neutrophils right here in this rightmost yellow circle. You can actually see that they have many lobes to their nucleus. Those are polymorphonucleocytes, remember? And we're also seeing those balloon hepatocytes circled in blue with some Mallory bodies again forming. Those are represented by the black arrows in this picture. Looking at the liver, very, very close high mag image of a ballooned hepatocyte with a Mallory body forming inside it. And that Mallory body, it's important for you to know that that's a glob of cytokeratin, basically cytoskeletal elements. It's just trash inside the cell, but it aggregates in clumps and gives you this characteristic finding of, again, an itis, not a steatosis, a hepatitis. And it's kind of twisted like rope or shaped in a little serpentine manner like a snake would be. It's bendy. There's a Mallory body one more time inside a ballooned hepatocyte. So all of those specific changes, or what are they specific for? An itis, an active, acute inflammatory response within the liver, not just benign fatty change. So in alcoholic liver disease, you're going to see a micronodular pattern of disease. That might be something to keep a running memory bank of, or maybe make yourself a list, because it's really low yield, but potentially still something they could ask you. Risk factors: men tend to drink more. Asians have this alcohol dehydrogenase variant that makes them alcohol intolerant. They don't metabolize it as well, so they get drunk off of 2 or 3 drinks instead of 20 like me. So hey, I guess that's not necessarily the worst thing in the world, like one shot and you're through the roof. So pathogenesis, this is where we start to go into the biochem side of things. And I want to start our biochem adventure by showing you a slide that is not necessarily taught by the Jedi. This is what's going on whenever you're interpreting liver labs, but I feel like all of our classes, this isn't really hammered home in any of our classes. So you see high ALT, AST. What that means is that there's immediate hepatocyte damage, and that's referred to as a hepatitic pattern. Obviously, hepa, liver, itic, inflammatory. So you see those two enzymes high, the liver's damaged. Alkaline phosphatase and gamma-glutamyl transferase. Now, you got a little ALP on the canalicular side of your hepatocytes. So literally, on the side of the hepatocyte facing the bile canaliculus, you've got ALK phos. And on the side of the hepatocyte facing the bile canaliculus, you've got a little bit of membrane-localized gamma-glutamyl transferase. It's a membrane-bound protein. However, where these things really are, I mean where they are just abundant, is within cholangiocytes. So you see high gamma-glutamyl transferase in the blood, barring alcoholic hepatitis. That's the only real liver disease that can make those go high. Anything else you see high GGT, that is a biliary tree disease. Same thing with ALK phos. You see ALK phos, okay, maybe the liver's involved, but really it's a biliary tree problem that you're thinking. Or maybe it's a primary hepatic pathology that is just spread downwards distally to the biliary tree. So could really, if you see all 4 elevated, you just know there's a problem right about there on the patient. It doesn't tell you anything really specific, but that's where these enzymes come from. And specifically, they pop out of these cells in response to damage. So when there's hypoxia, your hepatocytes are going to bleb, which means they're going to let go of parts of themselves, similarly to how a sinking ship wants to throw all cargo overboard that it can spare to lighten its load. Hepatocytes want to energetically make themselves more efficient. They want to let go of everything they don't need, and so they downsize, Like it's a recession. We're buying a smaller house. Does that ever happen to you? It's like, so they bleb and they let go of bundles of cytoplasm, and all of these enzymes are inside that cytoplasm. So that's what's going on. Now, one final point about ALT versus AST. AST is actually a very handy acute phase marker because there's more AST inside hepatocytes as a function of quantity than there is ALT. So in the very acute stages of liver damage, we're talking about like the first 4 hours. AST is going to be higher than ALT just by a bit. But then after that initial little spike, AST gets metabolized quicker than ALT does. And so ALT will be higher in the blood from that point on out. And especially in chronic processes. If you've got a chronic liver disease, maybe you've got multiple attacks of some pathology, well, that AST just gets metabolized, again, more rapidly than the ALT. So AST is going to be closer to normal than ALT, which is usually going to be higher, except in alcoholic liver disease. That's the one exception that you're thinking AST up. So that's our rationale for why a patient's going to show up, and have an ALT that's 65 and an AST that's 33, metabolized at different rates. Speaking of metabolism, let's talk about ethanol. There are 2 ways you can get rid of ethanol once you ingest it. And it's a real simple 2-carbon molecule with an alcohol at the end. It's nothing your body can't handle. It's got an enzyme that's really, really suited to breaking ethanol down into acetaldehyde. That's its only job. It's called alcohol dehydrogenase. And when it does so, it produces an NADH. And that's your cytoplasmic route of alcohol metabolism. And if you ever hear of the microsomal metabolism of alcohol, that's your cytochrome P450 system, your 2E1 variant, your microsomal inducible ethanol oxidation system. So that's going to make water. And also free radicals. Anytime you run a CYP enzyme, you have a small 1, 2, 3% chance of generating an oxygen radical. Very tiny, but even just normal functioning CYPs are kind of non-specific and haphazard. They just grab whatever it is they're detoxifying, and they grab an oxygen and they just smash that oxygen. Then they grab another one and smash the oxygen on it, and grab another one and detoxify it, grab another one and detoxify it. And a CYP enzyme is kind of like a monkey, like clapping the cymbals. You know, if you've ever like seen a wind up monkey, that's your CYP enzyme. That's how fast it's going. So because it's going so fast with whatever it's working on, and it's non-specific, it's got a loose binding site. Ethanol doesn't have to be snug in there before the reaction can take place. It'll just kind of go whenever ethanol gets anywhere close to it. That's why you have this potential to generate radicals even through normal physiologic CYP activity. So back to the NADH, that's really where we're going. You take acetaldehyde down to acetone, and you make another NADH when you do that. So you get 2 NADH every time you metabolize ethanol down all the way. Now, acetaldehyde and acetone have some serious implications here. Let's talk about acetaldehyde. It likes to grab onto proteins. And when acetaldehyde grabs onto a protein, it does so pretty strongly. It can't get off of that protein. And the reason acetaldehyde likes to do this is because it is an electrophile. And what that means is that it has a very electron-sparse region on this carbonyl carbon here. And so anything with extra electrons, proteins have a lot of polar moieties that can donate electrons, just might be able to grab this acetaldehyde and latch onto it. And the acetaldehyde doesn't care. It's going anywhere you got electrons. So that's how an adduct forms. And adducts can form with proteins, they can form with lipids, and they can form with DNA. And the fact that they form with lipids is also no small deal because well, you've got all these lipids that are floating around because all the NADH in the cell and all of the reactive oxygen species that are produced by your CYP2E1 enzyme, those ROS species can also peroxidate lipids. And you can quench that a little bit with glutathione and with vitamin E, but when you run out of antioxidants, well, then you've got a lot of damaged lipids out there. And the protein and the lipid damage is really what contributes to hepatocyte injury and the release of cytokines that gets the inflammatory response, those immune cells involved, that leads to cirrhosis over time. So all those radicals have a whole lot of targets in the liver because the liver is a very metabolic organ, tying it back to the title of this SDL. And I want you to keep in mind there's a big hepatocellular carcinoma risk at the end of cirrhosis. In fact, 70 to 90% cases of HCC have cirrhosis. You're not going to see a test question with hepatocellular carcinoma in which your patient doesn't have cirrhosis just about. Back to the NADH. Here we see it again. This is in first aid. So you need to know this, you know, because you might not need to know it for this exam, but it's in first aid for a great reason. What does that NADH do? Where does it go? Well, one of the first things it does is it totally blocks the conversion of pyruvate to acetyl-CoA through the enzyme pyruvate dehydrogenase. That is an NADH-dependent enzyme. And anytime you see a lot of NADH building up in the cell, because of all this alcohol metabolism, you see this NADH go up, 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 it's going to block the transformation of pyruvate to acetyl-CoA. So you can't go into the TCA cycle. You can't do aerobic metabolism. Well, what does that mean? It means that if you want ATP as a cell, you have to send glucose down to pyruvate through glycolysis. And then you have to send that pyruvate to lactate to get some ATP further out of it, because that's also an NADH-dependent reaction, and that's going to go to lactate. So that will create a local lactic acidosis, not good for cells, especially if it's constant, as in alcoholism. Number two, the way you can remember which way does this go? Okay, I'm going to teach you something right here. The way that you remember where the pyruvate, lactate, acetyl-CoA triangle of reactions, which way they're going, the side of the reaction that has the NADH is the longer word. So pyruvate, that has more letters than lactate. So when you think about which side is the NADH on of this reaction, it's on the side of pyruvate because that's a longer word, just like NADH plus H plus is longer than NAD plus. Easy money right there. Acetyl-CoA, okay, pyruvate, 8 letters. Acetyl-CoA, 9 letters, and a hyphen for free. So what side of the reaction is the NADH on? It's a longer word. It's longer than NAD+. So it's on the side of the longer word, acetyl-CoA. So that's how you remember the NADH-dependent regulation of that triad of enzymes. Really important stuff. I mean, this is kind of the crown jewel of all of metabolism, this triangle of regulation right here, you know, because this controls aerobic or anaerobic. That's important. So speaking a little bit more about aerobic, you get a lot of NADH because that's what happens whenever you break down alcohol. So in reference to the Krebs cycle, the NADH inhibits isocitrate dehydrogenase, and it also puts a stone cold stop on alpha ketoglutarate dehydrogenase. So what does that mean? Well, that means that everything is going to proceed towards acetyl-CoA because it can't go backwards. There's no way absolutely it can go backwards, and citrate can't go forwards. Isocitrate can't go forwards, so it's going to go a little bit backwards to citrate, and you're going to end up with an accumulation of citrate inside mitochondria. What does that mean? Well, that citrate can do one thing just about, and that's participate in fatty acid synthesis. So citrate is going to go out the mitochondria. It's going to become acetyl-CoA in the cytoplasm, and it's going to go straight to fatty acid synthesis. How do you get this? That's how. How do you get steatosis? Citrate leaves the mitochondria and builds fatty acids. So citrate builds free fatty acids, but you also need one other thing to make true lipid droplets, and that's a glycerol backbone, because triglycerides are the preferred storage form of fat. So where do you get the triglycerides? Well, you get it from fructose usually. And so we'll talk about a little bit about this with NAFLD, non-alcoholic fatty liver, but all the high fructose corn syrup in the American diet. Well, that fructose goes straight to dihydroxyacetone phosphate, which is very easily converted to glyceraldehyde 3-phosphate in the middle of the glycolytic pathway. And where is that going to go when you're in the fed state? That's going to glycerol, and that glycerol is going to complex with the citrate that makes free fatty acids. It'll complex with 3 free fatty acids, and there's your triglyceride. So that's fatty change in the liver. Going back to alcohol metabolism, specifically in the alcohol, in the fed state in general, but really in alcohol, you've got a ton of NADH. You've got so much NADH, and it's making citrate, but not only that, it's also making acetyl-CoA. And that is important because acetyl-CoA can bond with itself and kind of polymerize in a brief manner and make acetoacetate, which is the precursor for ketone bodies. So, you ever get a question about an alcoholic with a ketone body in the blood? They're in alcoholic ketosis. And that's going to be a ketoacidosis too, low pH. So the lactic acidosis, a little bit of lactate involved in that, a little bit of beta hydroxybutyrate involved in that. Why? Because this is an NADH dependent reaction. So if you've got a ton of NADH, you know, you don't need to make more NADH. You're sending it straight to beta hydroxybutyrate. So that's another mechanism. And then finally, we end up at the electron transport chain, and alcohol's got many important effects here. Number one, it's going to uncouple the electron transport chain, and it's going to do that because you're making more protons than you need, period. Why are you making more protons? Because you're dropping off more NADH at complex 1, and so your reductive potential is even greater than it ever was before you were drinking, you know? So all of that NADH gets turned into protons, but the problem is you do not need all of those protons to go to ATP. And so because you have excess protons, you have more protons than it takes to make the ATP you need, your body upregulates uncoupling proteins and sends them to the mitochondrial matrix. And that's represented right here on the right side of this graphic. And so the uncoupling brings protons back into the matrix, and that generates heat. This is why alcoholics, especially if they're, 12 drinks in and they show up to the emergency department, they're often running a fever. Might be infection, probably just alcohol-induced hyperthermia, makes you hot, I remember being in Cleveland, Ohio for college, and it's like we'd be outside in February and there'd be snow on the ground and you just see like 4 dudes walking by in like Carmelo Anthony jerseys, right? Why? Because they were skunk drunk and the alcohol was giving them heat. So something else is that this running of the electron transport chain gradually generates oxygen radicals. This is another source of free radicals, superoxide anion into the hydroxide radical, because complex 1 and complex 3 just naturally make them, again, 1%, 2% of the time you send NADH through, it's going to make an oxygen radical. And so normally you have enough glutathione peroxidase, vitamin C, whatever, antioxidants in general to quench these radicals. But when you are drinking, there is a critical mass leveraged against your antioxidant systems, because all of that acetaldehyde and ETOH that goes through the microsomal pathway through CYP2E1, that's another source of radicals. So you're going to be antioxidant depleted, and the 2E1 plus the electron transport chain, again, the radicals bounce off fats. There's a fat radical. Protein, there's a protein radical. So that's the mechanism of the damage here. And I mean, you could get into even more in detail questions about this. Okay, so I won't talk about it. SDL says everything that we just talked about with those diagrams. You make a lot of NADH, alcohol metabolism. The NADH turns on lipid synthesis, shuts down the TCA cycle. Pyruvate goes to lactate. There's your lactic acidosis. Excess NADH drives the G3P shuttle. Make that glycerol backbone. There's your triglyceride synthesis. So you know everything that this SDL is talking about like the back of your hand right now. How does protein synthesis get suppressed if they want to throw this at you? You're going to catch it like Randy Moss, and you're going to say EIF2 alpha, initiation of elongation initiation factor 2 alpha. There we go. Mechanisms of hepatitis: acetaldehyde adducts, radical formation, 2E1, electron transport chain. Those species make things really hard for hepatocytes. Now, because alcohol is depleting your antioxidants, it's going to deplete your vitamin C, your vitamin E, because alcohol is running. Let me show you this. This makes so much sense. Because this enzyme is a dehydrogenase. Tell me what B vitamin is a cofactor for every single dehydrogenase in the body. Thiamine. What's lactate dehydrogenase? Dehydrogenase. So what B vitamin do you need for lactate dehydrogenase? Thiamine. B1. Why do alcoholics have a B1 deficiency? There we go. So middle of page 5. Patients with cirrhosis have spider angiomas. If they could ask you something straight up as simple as that. Well, like, why does this patient have this angioma? And the answer is going to be they're peripherally vasodilated because a lot of nitric oxide that's specifically coming in from the gut and bypassing the liver circulation, making it out to the system. Gynecomastia, testicular atrophy, both those are due to failure to metabolize estrogen in adequate amounts. So you've got a lot of free estrogen floating around. Can cause breast growth unilaterally or bilaterally. Can cause, again, infertility even. Bottom of page 5, still talking about alcoholic liver disease. AST is going to be about twice ALT. Both are going to be elevated. That's particular. AST is localized inside mitochondria, and ALT is only a cytoplasmic enzyme. That's why AST is going to be higher in alcohol abuse. Again, remember, what did we just look at? Electron transport chain. Where is this? Mitochondria. Alcohol is sending a lot of NADH and generating a lot of oxygen radicals, as well as uncoupling that membrane. You're going to get leaky, dysfunctional mitochondria. And that's why you see a higher AST than ALT, because AST is inside the mitochondria. GGT is going to be up, not specific for alcoholic disease. You're going to see a macrocytic anemia. That's big cells, and that's going to be in chronic alcoholics, those with alcoholic steatohepatitis, so the active inflammation changes. And that macrocytosis means that they've got a B12 and or a folate deficiency. And so cells in their bone marrow, erythropoietic cells, have to grow, and divide. But whenever you have one of these two vitamins deficient, you can't make thymine, specifically that base T, right? So if you can't make enough T to divide, well, then you're just sitting there making proteins and growing, waiting for your chance to split in two and do some mitosis. And so when that finally happens, you know, when you finally have enough thymine to divide, you're just a bigger cell than you would be if you could have divided at a normal time. I hope that made sense. So that's why you get a macrocytic anemia with those deficiencies. Thrombocytopenia, alcohol inhibits the bone marrow. Megakaryocytes, which produce platelets, they just pinch off, they bleb off platelets essentially. Platelets are bags of cytoplasm from megakaryocytes. Those live in the bone marrow. Your INR is going to be up. That's secondary to failure of liver function. Prothrombin time is dependent upon clotting factors produced, synthesized by hepatocytes. And when we say clotting factors, what do we mean? We mean like factor 2, factor 4, factor 7, protein C, protein S, everything involved in hemostasis except for factor 8 and von Willebrand factor is getting made inside the liver. So if you get asked, why is INR increased? Well, it's because there's hepatocyte damage. Albumin level is going to be down. That's important. Why? Slowly, that participates in generating ascites, acidic fluid, transidative acidic fluid. So it's going to have a higher protein ratio than the surrounding blood. What's that? The SAAG, it's going to have a SAAG over 1.1. So what's the treatment for alcoholic liver disease? This is where we will wrap it up. It's abstinence, which you're not going to get that out of your patients 9.5 times out of 10, because alcohol withdrawal is one of the toughest withdrawals on this planet. Alcohol is a GABA channel agonist, and so it calms you down, because GABA is kind of a inhibitory neuron, right? An inhibitory neurotransmitter, and alcohol itself is a depressant. Well, that's why it works on GABA channels. GABA is a chloride channel, remember? And so if you are a alcoholic and you are accustomed to 12, 14, 20 drinks a day, then how much GABAergic neurons do you have in your brain? Yeah, so imagine taking all the alcohol away, and all of those GABAergic neurons are suddenly left there to itch. And so your brain's built up steadily these inhibitory circuits, but all of a sudden the circuits that should be inhibiting behaviors and moods and emotions and actions and physiologic functions, that off switch is suddenly turned on because you don't have the proper key to that off door. You don't have the stimulation of those GABA receptors. So what do patients with alcohol withdrawal look like? Oh, they're itching, they're jittery, they're sweating, they're swearing. It's a bad situation. It's tough to get out of there, and you might even have a seizure because your brain is so used to having this artificial dampening of its signal that when you pull that away, it goes haywire and it spikes with activity. It explodes, and then, yeah, that can absolutely cause a seizure, which could potentially be lethal. So alcohol withdrawal is absolutely no joke. It's tough to treat clinically. We have disulfiram, which does a fairly good job of keeping people from consuming alcohol. But the problem is we treat these patients and we put them back in the same exact environment that they came from that encouraged them to develop an alcohol addiction in the first place. So this is a tough disease. Non-alcoholic fatty liver disease. First thing that we said, this looks exactly the same as alcoholic fatty liver disease. Same progression, same steps, same result, same start, same finish. Only difference is there's no alcohol involved. So what is involved? What's the risk factor? How do you get this? Well, you eat like really bad and you get metabolic syndrome, which is defined as obesity and high triglycerides with low HDL, high blood pressure, and diabetes. But you don't need all 5 of those. Just 3 of those would give you metabolic syndrome if you're past the particular cutoffs. I would say out of all of those, obesity is probably the number 1 because obesity predisposes you to the other 4 here, essentially. And a risk factor for non-alcoholic fatty liver disease that is not covered in this SDL is a high fructose diet. And I'm thinking, Fruit Loops. I'm thinking Capri Sun. I'm thinking all that crap that we used to eat when we were kids, but now we are grown. And so hopefully you eat some more grown foods now, like get some steak on your plate. And fructose does some despicable things to your liver. Let me show you what fructose does. I found this one study. Here's a rat liver after 8 weeks of a high fructose diet, and it is half fat. Really. Fructose, as we said, goes directly into glyceraldehyde 3-phosphate by means of dihydroxyacetone phosphate. It cannot enter metabolism any other way than to go to glyceraldehyde. And that's a problem because that's going to go straight to that glycerol backbone of triglycerides in the fed state. And see, if you were eating glucose, your cell could do more things with that glucose, and that glucose is going to be oriented straight down the glycolytic pathway rather than fructose, which has to go in at G3P. And glucose can get turned into, let's say, glycogen, right? You can take all that G6P and you can say, well, you have enough energy, let's make glycogen. Can't do that with fructose. You have to make G3P first. So some of that goes to fatty acid synthesis. Not only that, I found a cool study, pretty recent too, that found that fructose inhibits carnitine palmitoyltransferase 1A, which is an enzyme on the outer membrane of the mitochondria responsible for transporting fatty acids into the intermembrane space. And then remember, there's a CPT2 on the inner membrane that's going to take those fatty acids into the mitochondrial matrix. And when those long chain fatty acids are in there, or medium or short chain, they're going to get oxidized, metabolized through beta oxidation. Well, this study found that fructose directly suppresses that CPT1 gene that's responsible for kicking off fatty acid oxidation. Glucose, on the other hand, pure glucose, actually had no effect on that gene. So fructose stopped it in its tracks and glucose didn't do nothing. So fructose really, really bad. That's it for the risk factors. If you see a risk factor question, pray for me. I always get those wrong. Same pathology here. A lot of fatty change, and fat is a big target for radicals, and that generates oxidative stress when fat lipid radicals accumulate, and the oxidative stress produces cytokines, which draw in neutrophils. Then if it's chronic macrophages, lymphocytes, they say, hey, what's going on over here? There's a lot of TNF alpha, So that's the inflammation that happens over and over and over again. And then you get some TGF beta every time M2 macrophages pop up when that inflammatory response goes down and your cells chill out. And that leads to collagen and scarring. So steatosis, itis, immune cells, and cirrhosis. This picture at the middle of page 7 on the left side of the page, this is steatohepatitis. And so when we said look for immune cells, if you see immune cells inside a liver biopsy, you're looking at an itis, you're looking at inflammation. This picture is a good representation of roughly how many immune cells you would need to see in order to make the call, hey, this is an itis. As you can tell, they don't have to be all over the page. You just have to see a good handful of them. I would call that a good handful of immune cells. So can you see ballooning hepatocytes in NAFLD? Absolutely. That's what we're looking at here in the middle. You see a chain of inflammatory cells, small dense nuclei, tight cytoplasm, and then we're seeing some ballooned, pretty pale, translucent, see-through type hepatocytes. Maybe a little bit of Mallory bodies starting to form up there and that one on the top. And then on the right, that's a trichrome stain, so that means we'll be looking at a bunch of blue collagen right there in the middle of this section here. With relative acellularity, you might see some lymphocytes, macrophages, etc. within that collagen. Zone 3 in adults, zone 1 in kids. Really interesting. Zone 1 in kids. I wish I could tell you a little bit more about that, but I can't. Clinical presentation, same thing. I feel like I just read the same paragraph about 10 minutes ago. Lab studies, normal AST-ALT ratio. ALT is probably going to be a little bit higher because this is going to be a chronic problem for most patients. Steatosis into steatohepatitis, into cirrhosis, into possibly cancer. Same exact thing. Pathogenesis. I imagine this section is giving some people fits because they're like, oh, do I need to know this? Do I need to know this? And I'm here to tell you, you probably don't need to know this. Or you probably would just be able to guess if they really asked you, what was the third hit? You know, it's like, are you going to say obesity? Like, no way, yo. Like, that's for sure. Like one of the first predisposing factors. It's a risk factor, for crying out loud. So what you really want to know is that mitochondrial dysfunction, stuff starts to go wrong with the mitochondria, and that's kind of the point of no return. And that's where you start to see hepatitis, inflammation, which is really tough to control. It goes into cirrhosis at a higher rate than just a plain fatty liver goes into NASH. So take a couple looks at it, but again, it's nothing that you're not going to be able to put one and one together and make two on during the test. Toxic and drug-induced hepatitis. Talk about sections that give people fits. This one was giving me fits because I saw this table, and I am not a table guy, yo. No. So I saw this, and I was like, oh, really? Okay. So I'm not going to learn that. Instead, I'm going to learn the old list. I can do a list. A list is like a baby table, right? So I can do a list. So bottom of page 11 into page 12, we've got a list of things. And there's even some pictures in there. So I'm like, yes, I can handle this. And the list tells you with a word or two or three what the pattern of injury is. And then the list gives you, right after naming the pattern, things that could cause that pattern. So for example, acute hepatitis could be caused by NSAID toxicity, amoxicillin toxicity. So in my mind, I say, okay, easy question. Patient, too much amoxicillin. What's the pattern of liver pathology? And the answer is going to be acute hepatitis. And then other examples are going to be like macrovesicular steatosis, microvesicular steatosis, cholestatic injury, fulminant necrosis, you know, and it's like all four of those, if you understand what they're talking about, you're like, well, it has nothing to do with fat, and you're not going to see bile, and it's not going to be fulminant, you know, like everywhere. So acute hepatitis sounds about right. So that's how I'm approaching this page. You look at the pattern and you figure out what's associated with it. Microvesicular steatosis, tetracycline, Valpro, cholestatic injury, oral contraceptives, can be acute or chronic. So really, it's a little less scary than I thought it was when I first looked at it. Cholestatic liver disease is mentioned here at the top of page 11. And we're going to have elevated ALP and GGT because one of those first slides we took a look at said that this biliary pattern of injury down here at the bottom, that's cholestatic injury. And you've got injury to cholangiocytes, which are the cells lining your bile duct. So that's why you get ALP, GGT. Because remember, those cells have a lot more of those enzymes than hepatocytes do. Isoniazid causes elevated liver enzymes. Amoxicillin causes elevated bile enzymes. Just a lot of information stitched together haphazardly in this section. So my best bet is to look at this again the morning of the exam and do a good thorough last pass with a highlighter and a paper SDL, you know. So that way, you know, I'm not going to remember it forever, but I'll at least recognize it if it pops up on the test. Let me see if I got some images for a couple of those different patterns. Thinking about acute hepatitis. Was that one of them? Yeah, so acute hepatitis is characterized by portal and intralobular inflammation and hepatocyte necrosis. So what we're really looking for there is going to be an inflammatory response. Here you go. This is fulminant hepatitis. This is an example of massive hepatic necrosis. And this is the sort of thing they're talking about when they say portal inflammation. Because, well, what's this right here? What are we looking at? We're looking at a portal triad. Here's our bile duct. Here is our arteriole. Here's our vein. And so you're seeing nothing but immune cells around this sucker. And the hepatocytes that you do see a little bit farther out have very large nuclei and tight cytoplasm. There's something sick with them. And you're also seeing a lot of hemorrhage in this photo too. So that's three great signs right there of acute hepatocyte necrosis, bad to the point of death, not just fatty change. I mean, bang, bang, bang. And how do you know? All the immune cells, all the red blood cells, something's up in that tissue. Same thing here, red blood cells, left lower quadrant, immune cells all throughout there. It's around a portal triad. So again, all right, picking up on, is it around a portal triad or a central vein? Every time you see that in an SDL, make a note of it. A lot of inflammatory cells in there. And then I want to look at a cholestatic pattern of injury. So this 6 and 7 on that list said acute and chronic cholestatic injury. What does that look like? Well, you see bile pooling in canaliculi in between hepatocytes. Sometimes you can even see bile pool within hepatocytes. The bile can aggregate to the point where it forms a plug, as I've highlighted in yellow here. That's going to lead to bile stasis and eventually backup and a hepatic jaundice. So that's a cholestatic pattern of hepatitis. Because not only do you have the cholestasis, if you'll also notice, you know, you got a lot of fibrosis going on here too, with many, many immune cells within that fibrotic infiltrate. So multiple things can happen on top of one another. Know the vocabulary, really understand what's going on with that cellular interaction vocabulary as best as you can. Another example of cholestasis, it can be spread out. And what's this associated with? Oral contraceptives, carbamazepine, anabolic steroids, a couple of things you could randomly get thrown in a test question. What you really need to know is acetaminophen intoxication. I mean, for sure know this because this shows up in the cirrhosis SDL as well. The big idea here is that whenever you metabolize acetaminophen, most of it gets glucuronidated, but some of it goes through the CYP system, makes NAPQI, which is toxic and must be immediately conjugated by glutathione. Now, problem is you take too much acetaminophen, you've got too much NAPQ, that overloads your glutathione and eventually depletes it, takes it out of the picture. So NAPQ accumulates and forms adducts with proteins. We know how that happens. And bang, fulminant liver toxicity. And how does this present? Well, ALT, ASTs in the 10,000s, very stupidly high. And you're going to see a zone 3 pattern of centrilobular necrosis around the central vein, and it's going to be bad. These are going to be discolored hepatocytes, a lot of a neutrophil infiltrate here because it just happened, you know. NAPQ specifically depletes the cell of calcium, and calcium homeostasis knocks your caspase cascade into action, right? 3, 8, and 9. But maybe the cell dies before you can even activate your caspases. That'd be due to membrane permeability and loss of isoosmolality, and so that's necrosis right there. How do we treat this? N-acetylcysteine, what does it do? It replenishes glutathione. It makes more glutathione so that we can get that NAPQ off of our liver. Okay, alpha-1 antitrypsin deficiency. Alpha-1 antitrypsin is a serine protease inhibitor. So protease inhibitor is the PI in these genotypes down here. We've got PIMM, that's normal. We've got PIZZ, that is worst case scenario, okay? And so this is codominant, which means there's more than one bad possible allele. So you could have an S allele and have the disease, or you could have a Z allele and have the disease. You can have any combination of M, which is normal, and either of the codominant alleles. So Z is worse than S. Proven because you don't get the liver disease, according to this chart, unless you have the Z allele. Okay, so that narrows it down. You get 5 alleles, 5 genotypes as an answer choice. Patient's got liver disease. It can only be 3 of them. So what's your next step? Well, okay, they give you serum A1 alpha trypsin, alpha-1 antitrypsin. And so then you're going to be looking at the concentration of that protein in the blood. And so let's say that the patient's got, you know, alpha-1 antitrypsin of 60. And then the question is, what's their genotype? You're going to say PISZ. Or maybe the question is, what's their risk of emphysema? And you're going to say 20 to 50% extra. So how does alpha-1 antitrypsin cause emphysema? Well, it's a protease inhibitor. A protease, what's that? It's a protein that cuts ACEs, other proteins. And where are some important protease inhibitors located? Well, neutrophils make elastase, and neutrophil elastase is very strong, and it say goodbye elastase. So where do you have elastase? In your lungs. Why? Because they need to spring back into shape after you breathe in. They need to be compliant and elastic. You get it, right? So if you don't have this protease inhibitor, neutrophil elastase just takes the alveoli of your lungs and turns them into Swiss cheese, you know, cuts them up like a second grade poster project. And where the liver comes back into play in all this is that you're going to see alpha-1 antitrypsin inside hepatocytes. It gets misfolded by the rough endoplasmic reticulum. It gets misfolded by the rough endoplasmic reticulum. And they're going to be PAS positive inclusion bodies. And you don't want to mistake these for Mallory bodies. No, don't do that. And don't mistake them for our Councilman bodies that are indicative of hepatitis. Don't do that either. You're going to see a lot of pink spots inside one balloon hepatocyte, and that's going to be your alpha-1 antitrypsin, because the patient will probably also have a cough or something like that, maybe a little bit of jaundice. This can progress towards cirrhosis. That's why I included this image, is because here you see nodularity with clear, extreme fibrosis in between nodules, you know, so it's like there's cirrhosis via alpha-1 antitrypsin. So put this on the short list of things that can cause cirrhosis and possibly hepatocellular carcinoma in a child. Because if you have the PIZZ type, right, you got to assume that that's going to present pretty soon if you have absolutely no protease inhibitor function. So absolutely requires transplantation. Only 10% of kids get the liver disease, but almost all of them with that PIZZ, again, get emphysema. And it's going to be a pan-acinar emphysema, meaning the entire acinus gets taken out, and it's going to be in your lower lobes as opposed to a smoker's emphysema, which is going to be a centralobular emphysema of the upper lobes. And you could have both of them at the same time, you know. Maybe you have one of these weak genotypes, like maybe you have PIMZ. And so the chart says you have no increased risk of emphysema, but if you smoke, you better believe you are going to get some emphysema like that, because not only do you smoke, you've got a weak protease inhibitor. So know the inclusion bodies, recognize the genotypes and the A1AT levels in the blood, and finally, accumulations of misfolded A1AT inside the endoplasmic reticulum of hepatocytes. So that's it for this lecture. In conclusion, big ideas. This pathway looks exactly the same for alcoholic and non-alcoholic. They really want to ask you about NADH producing fat because it turns on all those enzymes and it makes glycerol too. They really want to ask you about acetaminophen toxicity, like bank on it, count on it. So acetaminophen toxicity, zone 3 necrosis, really bad tissue death, necrosis, alcoholic and non-alcoholic fatty liver also going to start in zone 3, but that's not going to be like off of a waterfall necrosis. That's going to be a gradual, slower fatty change. Thanks very much for your time. Keep grinding, yo. You can do this. This exam is not going to be as hard as you think it's going to be because you're going to learn all of it before you take the test.